Thanks everyone for coming, and then thanks for the organizers for giving me the opportunity to share with you some of the more recent things that have come out of my group. And I'll let people know online, because we're going to be recording, that I do not want everything to freeze at any point here. So I'm going to go off camera while I'm presenting, because that sometimes causes a problem. So thank you all for taking the time to attend today, and I hope to be able to stimulate some thought on some of the new things that we've been doing, but they're still under the same theme of things you may have heard from my group before. And I want to do the most important thing first, which is acknowledge the people who did all the heavy lifting. So James Forder is a recently graduated PhD student, so former PhD, now at Merck. Hassan Shafar is a postdoc in my lab. I also did his PhD at UD in physics. And then Veer Palakolu was a postdoc in my lab and just recently joined NIMBLE. And for those who did log in early enough to see the picture in my background, it's actually from out of one of these windows up on I think it's this one right around here, if you can see my pointer. Actually, if someone can tell me, can you see my pointer? Otherwise, I will try and switch to a laser pointer or something that you can see better.
Yeah, we see it.
You see the pointer, okay, good.
Yes.
And then also acknowledge all the collaborators. So some of the work I'm going to show you was in collaboration with Bristol Myers Squibb, and some of it was in collaboration with Merck. And apologies for the typo. Let's see if I can get this to advance. There we go. All right. So I think for this audience, you're probably already well aware of this, so I'm not going to spend a lot of time to belabor the point that protein-based therapeutics are quite prominent nowadays in the global drug market. If anyone's interested in the timeline or a nice read about some of the history, there's a reference here in the bottom corner. The asterisks are next to, these are the top 10 just from this last review, that in terms of sales isn't always the best metric, but it is a metric. And an interesting anecdote is that if I had shown you this table 6 months before, or maybe even at the time that the COVID-19 pandemic hit, this would have been even more in terms of therapeutic proteins. But of course, given the timing of when this was published and the obvious things that have happened in the recent few years, there are also things that have to do with LNP-based, mRNA therapeutics. Though I would suggest, for those who are interested in getting into new areas, that a lot of the interesting physics that are underlie what I'm going to show today also can apply with some additional nuances if you think about the stability of LNP-based vaccines, as everyone had to live through if you had to deal with the fact that once the vaccine was thawed, you had 24 hours to use it or it was thrown away. But even though protein-based therapies have been around for over 2 decades and with any kind of prevalence, they still remain a challenge to develop. They're costly. We try and narrow the field as early as possible and try and pick your proverbial winner before even getting into phase 1 clinical trials, if you can. It's really not. So I come from small molecule background originally, and there it wasn't unusual to take more than one candidate into phase 1 clinical trials. That would be highly unlikely to try and do with a biomolecule at this point. It's just too expensive to move multiple molecules forward. And then we have the added nuance that these days it's expected that you should be able to administer with a small volume through a subcutaneous injection, or in some rare cases as an inhalable. But that does mean that you need to get a relatively high concentration on a mass per volume basis, because these are large molecules. And so you're trying to make those kinds of predictions with very limited material at the early stages, if at all possible. Okay, so there are a number of reasons that candidates might be problematic or even just fail or be left to the side. And some of those have to do with the manufacturing. And that has to do with lots of different things I'm not going to talk about today. But one of the ones that comes into play is the shelf life, and that does tie to the route of administration. And so problematic behaviors can be elevated viscosity. I'm not going to talk about viscosity much today. I'll just give you maybe one example, but definitely the protein-protein interactions. In this case, everything I'm going to talk about today is going to be self-interactions, so to speak, as in these are not mixed formulations of molecule 1 and molecule 2 of two different types. It's still interactions of molecules of the same chemical nature, chemical identity. But they can show elevated viscosity, liquid-liquid phase separation, opalescence, which you can think of as maybe an onset or an indicator of eventual phase separation, reversible self-association, which in this case I mean more like a dimer-trimer level, not a very large length scale level, and eventually even irreversible aggregation, which actually involves changes in the conformational state of the molecule. In the interest of time, I'm not going to really get into that last one much today. And so what's one piece that's common across all of that is the intermolecular interactions between proteins of the same species, as I was mentioning. And I think many in this audience would already be familiar with the fact that you can at least get a handle on some aspects of that with static light scattering or dynamic light scattering at low or high concentrations of proteins. The interpretation becomes a little bit more complicated at high concentration or a lot, depending on the system. And it isn't always true that net attractions will be problematic, but it's usually considered that it's likely that would be problematic. And often people are aiming for net repulsions with the anticipation that'll probably be more stable, with the assumption that what you want is the monomeric species at the end, that you're not looking for some sort of active dimer or the like, which occasionally happens. In some cases, these interactions, the measures of these interactions do correlate with what you see in terms of net viscosity at high concentrations. So high concentrations here would be on the order of 100 to 150 mg per mil. And so there's a good reason to think there's at least use in gathering this information or trying to predict this information at early stages. In some cases, and again, not all, the net interactions, this is not what people may be used to, not everyone may be used to thinking in terms of G22. It's the high concentration analog to B22, just with the opposite sign effectively. So in this case, repulsions are on the negative side and attractions are on the positive side. In some cases, the net rate of aggregation, just in terms of an initial rate or a rate coefficient for the initial slope of the rate of aggregation, does correlate with that. It doesn't always work that way. And I'm not going to talk, I don't really have time to talk about the irreversible aggregation piece today, but I can tell you that we've done a lot of work there. And even when it gets more complicated, the interaction piece still does tend to come out as being the self-interaction piece does tend to show itself as a key determining factor. So it's still useful to be able to get at this information, even if it is in a more complicated scenario. All right, so a lot of the work I'm going to tell you is from James Forder's thesis work, some of which has been published, and everything I'm going to show today has been published, and some of which hasn't. And I didn't have permission to share that just yet, so I'm not going to spend time there. But that'll be maybe for a future, come back for a future story. But the idea was to develop methods to characterize and predict self-interactions and ultimately aggregation in a net irreversible aggregation. And I'm going to tell you more about the first piece, which is what's on the left. And just to orient you, we use static light scattering to get after the second osmotic Vero coefficient B22. And for anyone in the audience who may not be familiar, you would typically measure the excess Rayleigh ratio, and you can, by plotting that or fitting that, I should say, as a function of the protein concentration C2, you can back out an apparent molecular weight, and you can back out the second osmotic virial coefficient. And you're ultimately gathering a parameter that captures a convolution of steric repulsions, electrostatic repulsions and attractions, and then a number of different contributors to short-range non-electrostatic attractions. And we can reasonably, at least with these types of protein systems, we can reasonably model already what the steric repulsions would be at this low concentration stage. And we have reasonable models for even the steric repulsions as a reference point at high concentrations. So you'll see that sometimes referred to. And those are really just using atomic level resolution models, but only capturing the sterics. So it's computationally efficient that you can actually do that these days. So you can use the steric value, which is what this B22 value on the y-axis is scaled by. So when you see the comma ST, it means we're using this steric only model, calculating that, and then using that as a scaling factor so that we can better parse out if you're above 1 in this type of plot, then basically the only forces that'll give you net repulsions in these systems relative to steric are electrostatics, and everything else is a mix of the other pieces. And then we typically do these measurements as a function of ionic strength so that we can get to high enough ionic strength to screen out the electrostatic contributions. And then we're really getting a lumped version of the van der Waals forces, hydrogen bonding, hydrophobic interactions, etc. And at higher concentrations, and again, C2 is the protein concentration here. If you extrapolate this out, basically what we're getting after is the 0Q structure factor with these size molecules with these types of light scattering measurements. And so we typically do all of this in the same instrument. And B22 is related to G22. It's just if you go to higher concentrations, if you see me plot G22, it's really just another way of representing the 0Q structure factor for the systems that we're looking at here. I think I'm going to, in the interest of time, I'm going to skip over this slide because I'm not going to show you any aggregation rate data in any detail. But if you typically in the industry, if you did want to look at aggregation rates, you would normally focus on the early stages. You wouldn't be watching monomer fractions drop down over multiple half-lives. You'd be focused on the first few percent loss, really. So that's when we think about shelf life, that's what you're normally thinking about. At least for ones that are going to be successful as products. All right, so I don't skip over some of the little animations. So when we're going to show you a few different types of data. When we're doing low concentration things, basically getting after B22, or as an example, we're going to be looking at 2 molecules in a box, and we're going to be enumerating this multidimensional integral, which is just from standard statistical mechanics. And we have a few different methods that do it efficiently. And that will have different kinds. I'm going to show you a few examples from different kinds of models. This just looks essentially 1 bead per domain. These are antibodies represented that way. I'll show you a few different kinds of models. When we go to higher concentrations, it's actually more efficient to use a grand canonical Monte Carlo. I'll see if the little movie will run. There we go. Which is the natural ensemble, if you want to directly measure the fluctuations, which are what really contribute to G22 or the 0Q structure factor. Static structure factor, sorry. So I can get the slide to advance. Okay. We try and use models that are based in physics, but have a minimal number of adjustable parameters. These are all going to be implicit solvent ones. So this is not just brute force MD, all the solvent molecules, all the co-solute molecules, all the proteins. That's just not viable at this point. So we're trying to balance between some reasonable facsimile of the real world and computational efficiency so we can actually get an answer and do something. And so essentially, I mentioned before that we have both electrostatic and non-electrostatic interactions. The short-range non-electrostatic interactions are in one lumped form in this model. And they end up having one adjustable parameter, which is the epsilon value for short-range attractions, essentially the well depth for something that looks like a modified Leonard-Jones potential. And that is what you would obtain by looking at the plateau value if you measure do these kinds of measurements and take them out to high enough ionic strength to screen out the electrostatics. And then the behavior when you go to low ionic strength will tell you whether or not you're getting net electrostatic repulsions or attractions. And then this is basically a screen to by function. And then there's one parameter, the psi value is the one that scales the fact that this is an implicit solvent. We're not tracking the counter ions. We're not tracking the fact that there may be Hofmeister effects. There may be preferential binding of ions to certain charge sites. We're not able to capture that. So we have a scaling parameter that helps us try and give some facsimile of that. And then we're going to look at a bunch of different models. I'm not going to show you the spherical cow, but we will actually look at the one bead per domain model, which for an antibody is kind of natural as a dodeca. So it's 12 because there are 12 natural domains, but also looking at things where we're going to start with, it's really one bead per amino acid. So each amino acid has a certain hydrophobicity, a certain size or diameter, a certain relative hydrophobicity, I should say, and a certain charge, +1, -1, 0. And then sometimes we compare to the all atom, but I'm not going to show the all atom ones today in the interest of time. We're also going to apply this to non-MAbs systems, or not monoclonals. So I'm going to show you some examples from a monovalent, these are fusion proteins, FC fusions, between a monovalent and a bivalent FC fusion, as well as just the simple fusion partner is just a globular protein that I'm not at liberty to say what its target is. That one goes beyond what we're approved to share. But all the details have been published in one of James's most recent papers. Give me a second. All right, sorry. So here's some examples from the FC fusion partner. And we see behavior where at 2 different pHs, we can switch from strongly net repulsive interactions. 5 to 7 times the steric value is really large. And the reason James plotted this as minus G22 is over 2 B22 is just that G22 and B22 are related by a factor of -2. But the numbers on the bottom, on the lower left, are so large that we're not actually measuring B22. We're actually measuring multi-protein interactions when we get to those kinds of conditions. So he was just trying to be a purist for that one. But you also see that the molecular weight, or the apparent molecular weight, under the net repulsive conditions is sort of what you'd expect. It's where we would think it would be. It's around that of what you expect for the monomer nominally. But with the higher pH conditions, we're getting sorry, I flipped that. At the higher pH conditions, as we get to high enough ionic strength, we're getting things where we expect the molecular weight for this size of a species. But at the other conditions, we're getting numbers that could be significantly higher. And when you simulate these, you get qualitatively and semi-quantitatively good results, but not quantitatively. And so the monovalent is a bit different. So here we get non-monotonic behavior in some cases. And ultimately at pH 6.5, we get essentially reasonably strong self-association, but I can tell you that it's reversible. And then when we simulate, we can capture some of this, but we don't always capture it completely. And it's more qualitative than quantitative. And I think I might have forgotten to mention, we're simulating this one with a 1 bead per amino acid representation. So basically the highest resolution that I'm going to show you today. And then with the bivalent, FC fusion, we get extensive oligomerization, particularly driven at pH 6.5. So you can see the apparent molecular weight is enormous compared to what I was showing you before. We do get the simulations to predict, for example, strongly attractive interactions, but again, only at 2-molecule level. The numbers on the left that's definitely more than just 2 molecules coming together. They're bigger than that. The oligomers are bigger than that. But at least at the 2-molecule level in the simulations, we're getting qualitative agreement from this level of modeling. So we said, all right, at least we're getting something out of this. We know we're not capturing everything quantitatively, but there's something going on where these are electrostatically mediated for reversible self-association. And so that's a common feature amongst the molecules that we had access to. And so James went about asking the question of, all right, can we at least, even though we're only getting qualitative, can we learn something that might tell us which parts of the molecule are responsible for this? So what I'm plotting for you here is that I'm only plotting so what we're doing is this is a 3-dimensional plot. So the z-axis, what's coming out of the screen towards you in terms of the colored red to blue, is the average interaction energy between residue I on molecule 1 and residue J on molecule 2 in units of kT. Right, so these are not large numbers necessarily, but they will add up when you add them up over all of the residues across 2 different partner proteins. And he's ordered them, so he's only ordered them in terms of the charged ones, because we did the ones with the non-charged ones and it doesn't change, so there's not much to show. But blue is net repulsive, so positive interaction energies, and red is net attractive. And what clearly jumps out as a pattern is, for example, with the monovalent, more so than the bivalent, is that it's the FC on molecule A, let's call it, with the partner or the fusion partner, the other domain of molecule B, and then it is symmetric. It has to be, because that's just the way these are designed. But you do see a mix, right? You see a mix of both positive and negative. Just may be that in some cases the negative is winning out to give you net attractions that is leading to the reversible self-association that we see. And there's a bit of blur when you get to the bivalent, and that may be because of the fact that there are 2 fusion partners. But clearly the FC is one of the bad actors here, which means if you want this type of format of protein, it's what you're going to have to live with. And if he zooms in on this one, on which residues you can try and pick those out, and I wouldn't be able to tell you off the top of my head which amino acids these were anyway, but I'm sure I'm not allowed to tell you those as well, since they would help to identify the fusion partner. But the most influential amino acids are similar for the different fusions when we go and check whether it's the monovalent or the bivalent. So that's story number 1, and shows you that if just with a minimal amount of experimental data through static light scattering in principle, one could use DLS too. In maybe a platelet format, you have to do a bit of a correlation between the DLS and SLS in order to be able to do that. But there's experimental data on that, at least for MAbs, that can be done. And then, so you can use these kinds of tools, and at least that was low concentration work, and you can get insight into at least tuning these kinds of computational approaches and gain insight into what's really driving the bad behavior or the unwanted behavior. And then that helps people go back and maybe redesign their proteins. And I'm going to change gears a little bit and now push more towards the high concentration side of things, where we're going to be looking at monoclonal antibodies. The prior work I was just showing you, there wasn't really an interest to go to high concentrations, and to be fair, the amount of material needed to go to high concentrations experimentally wasn't available. So here we're going to go back to the simpler system of monoclonal antibodies, but now push to high concentrations. And give me one second. Apologies, fighting a early spring cold or late winter cold. So I've already shown you some representations of this. I'll just reorient a bit. So we're going to use low concentration experiments, which will be, in our case, static light scattering to get at B22 values. We're going to do that as a function of ionic strength and pH for any given protein, and we're going to do a series of them, a series of different monoclonal antibodies. And that'll help us tune our model systems. Basically, it's effectively something to pin down how relatively strong are the electrostatic contributions compared to the non-electrostatic contributions. And we'll simulate the B22 values, use that to tune those model parameters, and then we'll input that into predicting the high concentration behavior. So we will not be fitting the high concentration behavior against the experiment. We're going to do a head-to-head comparison of blinded experiments and blinded simulations. So we kept those two separate, and then we see just how well could you do. So basically, can you take the physics from the low concentration side of things, apply it to the high concentration, and just increase the concentration in the simulations, and how well do you do? And then we're going to compare that across some different models to see whether or not it's really a test of what's the level of detail you need in the model in order to probably get this right. And we have experiments from some of James's work plus some prior work. So we're going to look at half a dozen different MAbs, different isotype, so it's basically either all IgG1 or IgG4. We're going to use different pH values, deliberately trying to cross over the histidine pKa, and also because that's what's industrially relevant. And we're going to look at a low ionic strength and a high ionic strength to not completely screen out the electrostatics in some cases, but mostly. And the data that we have went up to 130 mg per mil in these cases. Okay. And so to orient you, so now this is 0Q structure factor. So in this case, everything's supposed to go to 1 and does at the lowest concentrations. Again, x-axis is protein concentration. And so we find under some conditions, I'm going to just give a couple examples here. This happens to be MAb2 at the lower pH, pH 5, and in this case, it's low ionic strength, behaves in a typical colloidal sense. You basically, if you had sterics only, that's what the purple dashed line is. And so we get, this is even more repulsive than that. And so that's basically electrostatic repulsions, and it behaves effectively what you would expect from a colloidal model. And just showing you that the symbols are for doing a straight grand canonical ensemble simulation, and those are the experiments. And then the curve with the shaded area is from one version of how we do the simulations, such that you can just piece the whole thing together and you get a nice smooth function. In other cases, you'll see we run separate simulations to match the particular concentration. And so you'll see just different data points. But here with the dodeca, it's fast enough that you can just simulate effectively a continuous curve of concentrations. And so the domain level model works quite well. So you're basically capturing the net charge in each domain, and that's doing the job. You go to reasonably attractive conditions. In this case, you look at the static structure factor, your numbers way above 1. In fact, we can't even get above about, I think, 35, 40 mg per mil in this case, and because it starts phase separating on us. So experimentally, we couldn't even go any higher. This is a different MAb, different pH. The purple dashed dot line is the same as it was that'll be the same for all the MAbs. And in this case, the dodeca simulation starts to show attractions at lower concentrations, but then falsely shows that it would become less attractive at higher concentrations. And effectively, it's because we're not capturing the right distribution of charges on the surface. So I'm going to show you a little bit of detail on what you might want to look at with a higher resolution model. And so the domain level models are great because they're computationally efficient. You but you're lumping multiple amino acids per bead, and so you're losing that spatial distribution. You'd love to go to the all atom, not computationally tractable, partly because the force fields that do all atom need you to also include all the solvent, and that's not going to work at high concentrations for what we're trying to do here anytime soon. Could try and go to the 1 bead per amino acid, which worked quite well at those low concentration behaviors for the fusion protein I was showing you before, but it's still really computationally burdensome because you're tracking a whole bunch of beads in here that you basically, it's 1,500 amino acids in here, and it's maybe usually a tenth of that are actually charged. And since the simulation times scale as the number of beads squared, it really hurts you in terms of what you're trying to do. So a merger of that is something that Hassan came up with in some of his earlier work, is to say, well, the sterics are still important. You need to have things in there. Then the non-electrostatic attractions have some role to play. But if really it's dominated by the charges, why not do sort of basically a hybrid? And so this is the 1 large bead per domain, the big gray ones, and then specifically pin down where the charged beads are and track those as well. And so it makes this tractable to go to high concentrations, yet still trying to capture at least the essence of the specific electrostatic pairwise interactions. So at conditions where it's net repulsive, one of the examples I was showing before, you could use either model. Each one works just as well, just the domain level model is just much faster, orders of magnitude faster. Or computationally burdensome is maybe the better way to put it. But the 1 BCD does just fine too. So when the 1 BCD, it is still significantly more expensive computationally than the domain level only. And so in that case, we don't run so many simulations that we could just splice them together as a smooth curve. And that's why you're seeing symbols there. We actually simulate to hit the same concentration as the experiment. When you go to attractive interactions, where the domain level only model would fail, we find good qualitative and in most cases quantitative agreement with the 1 BCD model. And again, these are not fits. These are using the low concentration information only, and then let the model try what it can do at high concentrations and what do you get. So these are true predictions of the high concentration behavior. When you get to really strongly attractive interactions, there I would say it's definitely not quantitatively in agreement, but we're semi-quantitatively in agreement, and you certainly have the right qualitative behavior when you go to the 1 BCD. So those were some successes. If I try and summarize what we saw across all of the different MAbs and pHs, and I'll say that many of the conditions that we looked at ended up experimentally being reasonably repulsive. In general, you get good agreement, and if you knew that you're looking for net repulsions, then the domain level model is just fine. But when you want to go to attractions, particularly strong attractions, it usually falls flat in its face. So I do want to leave time for questions. So to give you a high-level summary of the journey so far, is that if you are looking to use coarse-grained models for simulations of at least the types of therapeutic proteins that we've been able to play with, domain level models are great in some cases. They're fast. It literally can take minutes on a laptop. And they will capture the simple colloidal type electrostatic repulsive behavior that sometimes we see. But if you get to stronger attractions, and if you want to go to high concentrations, you've got to consider something a little bit more nuanced. And it does cost you speed, but it does, if you can do something that will allow you to track the specific sites for the charged species or charged residues, then it's doable. We found that it's tractable, and it's on par with speed or better than it would be to just do the experiments brute force yourself. So that's good. because it means that at least the computers is as good as taking the time to try and go in there and do it by hand yourself in an experiment. And it also means that if you want to do screening of many different molecules, now you can start parallelizing things and you can start moving more towards the discovery side of the world instead of more of where we started this journey probably 15, 20 years ago, which is that we almost always were looking in hindsight. At the time, this really wasn't viable from a discovery perspective. It was always wait and get the molecules you get, whatever industry can share with you, and then you're telling a story in hindsight as to why things behaved the way they did. Whereas here we're getting to a mode of we can start predicting with some confidence what the future would look like in terms of candidate selection. And so that's really a great success from James's work and the others who he collaborated with. And I think I already mentioned at high concentration, obviously this gets even more nuanced. So I think, yes, that's my last slide, is that I want to come back and do a big shout out for James. And most of what I showed was his work, but some of the underlying core pieces came from Hassan's work. And some of this was a collaboration with Veer. And then I'm not going to read off all the names, but we have multiple collaborators across both BMS and Merck & Co. And then I'll always have to acknowledge, generous support of BMS and Merck both for the portions of that they funded, as well as the Biomolecular Interaction Technology Center, which is an industry consortium that helps to fund kickoff projects within this space.